For those of you who are thinking of doing research here at UofM and are looking for a director for your broader impacts and NSF proposals, the conservatory alone has over 120,000 public visitors per year. We're actually the highest public engagement element of the university, other than the hospital and athletics. So we're very able and willing to help you with your broader impacts. So I'm going to turn this mic back over to our team. Perfect. Then we're going to turn off slides.
Okay, we're going to wait just one more minute. And then
I think you have to use that.
I think this
This is working.
is working now. Maybe. You can see it. Maybe okay, so we'll get going here. So it's my pleasure to introduce Meredith Zettlemoyer. Did I say it right? Zettlemoyer. Zettlemoyer. Meredith did her PhD at Michigan State University with Jen, our first year. So it's like a mini reunion happening, which is very exciting. And then now is doing a postdoc at the University of Georgia. And Meredith sort of integrates approaches from ecology and evolutionary biology to study species interactions and how they change across the globe. And we're really excited to have her here. So here's your goodie bag. Thank you, Meredith.
Thank you, everyone. It's really exciting to be here. I attended this symposium at least twice as a graduate student, so it's really neat to kind of be on the flip side to get to tell you all a little bit about some of my work on the consequences of phenological shifts for performance at a couple different biological scales. So shifting phenology, or the timing of life history events, is one of the most common responses to environmental change. So we see patterns such as advancing migration in birds, and then advanced or compressed breeding across a whole variety of taxa, including in plants. And so for plants, the timing, the life history transitions that we commonly talk about are germination, leaf out, flowering, and fruiting. And the timing of each of those transitions is often sensitive to local environmental conditions, such as temperature or precipitation. And so as climate change alters local conditions, there may be this mismatch between the conditions that have historically seen an organism that's a good time to start growing or reproducing versus the conditions that they're experiencing now already and then into the future. And generally, we know that phenology is advancing with warming climates. So in this meta-analysis of spring phenological trends, most species are advancing their phenology over time, but there are a handful of species that either don't change their phenology at zero or that are in fact delaying their phenology. So these shifts are advancing, but pretty variable under warming conditions. And shifting phenology can have a whole variety of different consequences. So it determines exposure to environmental conditions. So for example, if you're a plant, you flower too early in the season, you might get hit by these late frost events. But on the flip side of that, if you flower too late, you're going to get hit by these summer droughts. It also determines access to resources like space, water, and light. And it affects species interactions in the community, including between plants and their pollinators, as well as competition with other species, including invasive species. So altogether, shifting phenology is really likely to influence both performance and, at a broader scale, species success. And indeed, we do see that earlier phenology is often associated with higher reproduction. So in this native annual from Europe, Dittrichia, Relative fitness or seed set is higher when flowering day is relatively early in the season, and then fitness declines as flowering day is later. And then phenology is also associated with both increases and decreases in abundance in natural populations. So in this study out of Thoreau's Woods in Massachusetts, researchers monitor changes in both abundance and flowering time over several decades using historical records. And they found that species that were experiencing major declines in abundance, so this is going to be any of the species shown in red on the nodes on this phylogenetic tree, were species that didn't really shift their phenology over that same time period. But the species that did manage to increase in abundance did generally advance their flowering time and were these more successful exotic species within the community. And today I'm going to be telling you about the impacts of shifting phenology across 3 biological scales. So I'm going to talk first about individual variability in plastic or phenologically plastic responses to climate change. Then I'll address how shifting phenology can potentially affect population level fitness, and then how differences in phenological responses between taxa that vary in how successful or widespread they are can affect these macroecological patterns of both invasion and extinction. Okay, so getting started with individual variability. So most studies that think about whether phenology is shifting in response to climate, so here I'll be showing you a series of figures showing this differences between historical and forecasted climates. These studies typically look at whether phenology is shifting at the population level, so this black bar here. But this approach assumes that every individual within the population, so each of these gray lines, is shifting their phenology in both the same direction and to about the same degree as that overall population mean. But individuals can vary quite a bit in their responsiveness to climate or demonstrate these individual by environment interactions. And so in this case, we might have some individuals that either shift their phenology in the opposite direction as the population mean, or they just vary in the magnitude of their response. So here we have some individuals that don't demonstrate a massive shift in their phenology under warming, whereas we have others that are very responsive. So they show this steep slope in response to warming conditions. And I by E can result in different amounts of variation in phenology, where under historical climates, we might see that individuals largely have very overlapping phenological timing, whereas under forecasted climates, there's going to be this wider amount of spread in the phenological events within a population. However, we have really limited evidence for I by E in natural populations, especially in plants, simply because it's very data intensive. You need to monitor the same trait on the exact same tagged individuals over several years. So in my postdoc, I'm working with a group that's been monitoring phenology of Silenia caulis, which is a really cute cushion plant, alpine cushion plant that grows out of the ground in tundra above tree line, in a long-term observational study based at the Niwot Ridge LTER in Colorado. And so over the past 6 years, we've collected data on flowering time and fruit production on the same tagged individuals, and are looking at their responses to temperature, growing degree days or GDD, as well as snowmelt timing. So in this system, unsurprisingly, flowering time is advancing with warming. So this is showing you first flower as day of year in response to the number of growing degree days accumulated in a given growing season. And so flowering time is earlier when conditions are warmer. And similarly for snowmelt, we see that flowering time is earlier when snowmelt is early in the year. So we're looking at I by E in Silene because it's an extremely long-lived perennial. It can live for more than 200 years. And so phenological plasticity or plasticity in general is really likely to determine whether it's going to persist or potentially go extinct under climate change. So looking at individual Silene plants, I'll first address whether those individuals vary in their phenological responses to climate. And then second, how variance in phenology is shifting with warming conditions. So here what you're seeing is individual reaction norms showing the first flower response to GDD for every tagged plant in our study. So we quantify these using random slopes. And so what we can see here is that most individuals are flowering earlier when conditions are warmer, but there are individuals that delay their flowering, and there's a lot of variation in the magnitude of responses among these individuals. And we see really similar patterns for snowmelt, where flowering is earlier when snowmelt is early in the year, but again, there's a lot of variation in the direction and degree of responses. So the next thing we did was try to simulate how that variance in flowering time was shifting with warming. And as we expected, we found that individual variance in first flowering time increased with warming conditions. And similarly was higher when snowmelt was earlier, with the exception of one site. So our sites are arrayed to essentially capture enough variation in snowmelt timing across the study. So the data here is like a bit extrapolated. So this first section, we do see that individuals are generally advancing their flowering, but to quite different degrees. And that variance in flowering time is increasing with warming conditions. And so some of the next steps for this project are to try to link those individual reaction norms or those individual slopes with our measures of fitness to really test whether phenological plasticity is adaptive in this system. So next we'll think about effects on population fitness. So here I'm showing you 3 different scenarios for shifting phenology in declining, stable, and expanding populations. And so what we might expect is that in the declining population, we might not see that much change in phenology under forecasted climates. In the case of a stable population, what we would expect to see is that the population is essentially shifting its phenology just enough to track suitable environmental conditions in time. And then in an expanding population, we might expect that there's this greater phenological shift or steeper slope in phenology between forecasted and historical climates. And these scenarios might correlate with changes in population growth rates. So here I'm showing you the same 3 scenarios corresponding with changes in lambda or population growth, where the cooler colors indicate a declining population, orange indicates a stable population. So this is close to lambda 1, the population's at replacement, and then red indicates a growing population where lambda is greater than 1. And so in a population that doesn't shift its phenology, we might see this decline in population growth rates under forecasted climates. In a stable population, we expect that phenology shifts enough to basically maintain population growth. And then in an expanding population, we might expect that there's some benefit to being able to advance your flowering time enough that increases population growth rates to be greater than 1. And so now thinking about populations of Silene, I'll address whether phenology is under selection, and then second, whether phenological plasticity is potentially adaptive by tracking this sort of optimum phenology with respect to climate. So to do this, we tested for selection on 2 climate relevant measures of phenology, kind of from the plant's perspective. So to do this, we used fruit production per unit area as a proxy for fitness. Lambda is sensitive to reproduction in the system, so it's a pretty good proxy. And related this to 2 climate relevant measures of phenology. So first, the number of growing degree days that are accumulated from the start of the growing season to the time that a plant produces its first flower. And then the number of days between that day of snowmelt when we estimate that there's no snow left on the ground and the production of that first flower. And so in both of these cases, lower values indicate that flowering is occurring under cold conditions. And the caveat here is that that can occur in 2 different ways. It can either happen because an individual flowered very early, so with very few GDD accumulated or very quickly following snowmelt, or simply because that was a data point in a very cold year on average. So that's kind of the caveat with this particular measure. But what we found was that fruit production is highest at very low values of both GDD as well as days since snowmelt at first flower, indicating potentially directional selection for flowering under colder conditions or flowering earlier, depending on how that happens. And so we then tried to tease apart this 3-way relationship between climate, and here I'll only be showing you seasonal GDD, flowering time, and fruit production using non-parametric or nonlinear fitness landscapes. And so what we see is that reproductive success is highest when conditions are cooler. There you go. And it's also highest when flowering is kind of early to intermediate in the growing season. And so since we know that phenology is shifting to be earlier and appears to be under selection, to be under cold conditions, this indicates that phenological plasticity is tracking this phenotypic optimum to flower under these cold environmental conditions or earlier in the year. But we also see that fruit production declines really rapidly when conditions are really warm, even when flowering is quite early. And these are conditions that we are already seeing at our field site within the last 6 years. So this suggests that, yeah, it might be adaptive, but that might not be enough to maintain fitness as climates get warmer and warmer. And actually, if you do sort of track the populations over time, so these larger symbols are showing population means in each year. Populations are falling from these high fitness isocyclines in red and yellow into these lower fitness isocyclines in blue. So for this section, we see directional selection for early flowering or flowering under colder environmental conditions. And it appears that that plasticity is potentially adaptive, but that might not be enough to maintain fitness under warming. And so we're currently working on setting up some experimental manipulations to really try to tease apart the relationships between phenology, snowmelt timing, as well as interactions with pollinators and the co-flowering community at our site in Wet Ridge. So lastly, we'll talk about how differences in phenological responses can affect macroecology. So far I've shown you how these differences in phenological shifts can scale up to affect population patterns, but then those differences in population growth rates can then translate to differences in the spread of a species across a landscape. So here you're seeing historical versus forecasted distributions corresponding to the scenarios in these panels, and the colors in these circles correspond with the lambda values in this heat map. And so in a stable population, we expect to see no change between our historical and forecasted distributions. In the case of a range shift, we would expect population growth rates to remain about the same, and the population to simply migrate poleward to track suitable environmental conditions in space. In the case of a declining population that doesn't shift its phenology, we would expect to see population growth rates to decline, resulting in this range contraction, or potentially local extirpation. And then in the case of an expanding population, so for example, invasive species, we expect that advancing phenology increases population growth rates, and this allows the population to grow and spread across the landscape. So here we'll look at how differences in phenology could underlie invasion and extinction by comparing responsiveness between first native and non-native taxa, and then locally extinct and extant taxa. And we'll do this using two complementary approaches, so a field manipulation and then natural history records. So to look at invasion, I used a simulated warming array at Kellogg Biological Station. And so in this old field environment, we have these plots where half of them are left at ambient temperatures. And then those have sensors in them that raise the temperature of the warmed plots by 3 degrees Celsius, so about matching our regional predictions for warming. And the plots are warmed by these infrared heaters that essentially just shoot heat at the plants in the middle of these triangles. So into this array, Jen's group planted 50 species where the species were paired in the same plant family, so that one species in the pair was native in this top row, and it was paired with an exotic species in the same family as in the bottom row. And we monitored the number of days to first flower from the start of the growing season between these native and non-native species. And you'll see the ambient temperatures in blue and the elevated in orange. So what we found, first with the non-native species flower on average about a month earlier than the natives. And so this could potentially give them early access to those resources like space or light or pollinators, so they could potentially benefit from priority effects in that way. But we also found that the native species did not change their flowering time at all in response to warming. whereas the non-native species were able to advance their flowering under these elevated temperatures relative to the ambient ones. And so that could give them a huge advantage as climates continue to warm. And so we're currently working on compiling some of the growth and fitness data from this experiment to test whether some of those phenological differences actually contribute to demographic differences in success between the native and non-native species. So for invasion, we see that warming advances the flowering of these non-native, but not the native species. And then to look at differences in extinction, I've been examining extinction events in Kalamazoo County, which is where KBS is located. And so this area has really rich botanical records. It was surveyed in the early 1900s by Clarence and Florence Haynes. And their field notebooks were published as the first flora of the county in 1947. And then in the 1990s, a local undergraduate from Western Michigan went back out and resurveyed their sites and talked to local botanists and essentially got the second snapshot of diversity in the county. So between these two historical records, we have some idea of the presence and absence, but not abundance of species over that time period, and some information on the habitat types where they were found. So I use these records to try to figure out what type of species were most at risk of loss, and found that our native prairie specialists, so these are species that were only ever found in these threatened tallgrass prairies that have been mostly dug up for agriculture and development since, have been lost over that 155-year time period from this area of Michigan. And from those species, I then selected phylogenetic pairs in which each pair has a locally extinct species and it's paired with a species that's still extant or still persists in the landscape. And so this comparative approach has allowed me to test some of the differences in traits and responses to global change that might underlie the differences in success between these species independently of differences in their phylogeny. And so I worked with a great REU student at MSU, Katie Rinaldi, who was interested in whether differences in phenology could underlie some of these differences in species success. And so Katie quantified phenology on thousands of herbarium specimens and compiled local climate data. So she used the day of collection from the herbarium specimen label as a proxy for flowering time. And we examined the relationship between this day of year of collection or flowering time with several different climate variables, although today I'll only be telling you about mean spring temperature. And so Katie predicted that the locally extinct species, which I'm going to show you always in red, would not change their phenology in response to climate, whereas the extant species, which I'll always show you in gray, would advance their phenology in response to warming. And so what Katie found is that the extant species, so every line here represents an individual species. The extant species generally do advance their phenology in response to warming, whereas the locally extinct species just showed really variable responses. So some advanced flowering, some delayed, some didn't respond at all. But once you average across these really variable responses in these two groups, what we found was that the extant species were able to shift their phenology by about 5 days per degree Celsius, whereas the locally extinct species were only able as a group to advance by about a day and a half. So we've since planted some experimental populations of these species in the same warming array and are testing phenology's role in extirpation. So for extinction, we see that the locally extinct species show weaker, or at least more idiosyncratic phenological responses to warming. So today I've shared some work on the impacts of shifting phenology across these 3 biological scales, specifically looking at individual variability in plastic responses, how shifting phenology affects population fitness, and then how differences in responses can affect macroecology. And so I hope I've convinced you that it's important to consider responses across biological scales, because individual variability in responses to global change can affect population patterns, which might then translate to these differences at broader spatial scales. And with that, I'd like to thank the many people who have helped with all of these projects. as well as my research mentors and funding sources, and take any questions that you all might have. Thank you.
Good afternoon. As we're ready to start next session, a couple of comments about the posters and then we'll turn right on. Poster sessions begin at 3:30. For those of you who are in the poster session, we have many of your posters, some of you may have brought yours. We're going to bring the poster card in here. Don't leave the room, and we'll announce it at the end of the last keynote Q&A. Just don't go anywhere else, try and find the poster.
Okay, hello. Okay, so let's start. Right now, it's my greatest pleasure to introduce Dr. Perry Triggle. Dr. Perry Triggle is a postdoc and an NSF postdoc fellow at the University of Hawaii at Manoa, but also at the University of Kentucky. She earned her PhD in integrated biology at the University of California, Berkeley. And she's interested in using sophisticated statistical methods to model natural phenomena, particularly in plants, green life. And she does really cool macroevolutionary work in plants, and I'm really excited to see what she's going to present today. So yeah, join me to welcome Carrie.
All right, can you all hear me and stop picking up my voice? Awesome. All right, thank you so much, Faith, for that introduction, and thank you for to all of you for being here, and to all of the organizers of this symposium. It's been a really wonderful experience with me here, and all of the travel logistics were incredibly well planned. So, like, hats off to everyone involved. Like Tay said, I am a macroevolutionary biologist, and so I'm going to tell you today about some of my work exploring how below ground structures allow plants to thrive in seasonal climates. But before I get started, I just wanted to quickly acknowledge that their current graduate students and instructors are on strike, and so I appreciate that many graduate students here may feel torn in multiple directions right now. And so just know that sort of I'm aware of that and really appreciate the extent to which you're able to participate in this, and like we're thinking of you and your struggles in that way. I also wanted to say that I am currently working at the University of Hawaii at Manoa. I'm also originally from Hawaii, and the incredible levels of biodiversity in tropical systems really is my main motivation as a biologist. I seek to understand how this biodiversity evolved, and I'm also interested in how the current biodiversity crisis is playing out in tropical systems. I also wanted to say that my family and I are Haole, we're guests in Hawaii, and so just to acknowledge that the state of Hawaii was illegally, the kingdom was illegally overthrown by the US government, and that's sort of the history of the place that I currently live and work in. Not only is Hawaii an amazing place to sort of see this biodiversity and the processes that generate it, but it's also a really outstanding, in a bad way, example of the modern biodiversity crisis and ongoing rates of extinction. So this is a view from the tops of the tallest peak on Oahu, the island that I live on, Ka'ala, looking down into some of the lowland habitat. And so you can see from the peak of this mountain some of the most biodiverse and intact native forest habitats that remains in this island, looking down into the coastal areas where there's been significant agricultural and human development that's drastically altered native ecosystems. It's pretty rare to find native plants anywhere in that lowland habitat, except for some coastal areas. So that's the extent of biodiversity loss that's happening. Of course, these processes are not unique to Hawaii. Biodiversity loss is happening across the world, and as these forces are shaping biodiversity patterns, particularly evident in montane ecosystems, such as this region of Peru, where you can see that native habitat, because of is being pushed up against the limits of available land in these high alpine environments. And that's again being driven by human development and exacerbated by climate change. So my research, I'm really interested in how plants evolve in response to global change and disturbance. But when I say global change and disturbance and evolve, I mean over the course of millions of years. And so in this talk, I'm going to take you back on a much sort of larger timescale set of questions than what some of the other speakers have been talking about today. So thinking about the evolutionary history of lineages at a macroevolutionary scale. In particular, I'm interested in how and if we can identify and quantify the evolutionary forces that shape convergent growth forms in plant morphologies. And ultimately, I'm really interested in to what extent evolution is repeatable. And so when we see the repeated evolution of morphologies, do we also, is that happening through similar evolutionary mechanisms, and is that being shaped by similar environmental forces? In my research program, I try to answer this and other questions by integrating across 3 different main areas. The first is in characterizing novel biodiversity, which I do through fieldwork, work in herbaria or museums, as well as lab work. I also seek to develop nuanced models of evolutionary processes and develop software tools that allow for more user-friendly access to those types of models. And I see these 3 areas as really interacting. And so today, I'm going to tell you about a project that I think is a good illustration of the importance of integrating at least these 2 areas here, where we can see that the importance of understanding some of the nuances of biodiversity and of the actual fundamentals of organismal biology, and also bringing in more nuanced models of evolutionary processes to appropriately model that biodiversity that we see in natural systems. My background and my general approach is through the field of phylogenetic comparative methods, where we test macroevolutionary hypotheses using statistical models of evolutionary processes. I think of phylogenetic comparative methods as an inherently interdisciplinary approach because it allows us to leverage evolution, botany, genomics, mathematics, and statistics, and computer science, all in a single united analytical framework to test hypotheses, but bringing in information from these really different areas. Some of the questions that we can answer with phylogenetic comparative methods are if a morphological trait has evolved once or convergently multiple times, if suites of traits evolve together across a phylogeny, and what processes mediate biodiversity dynamics across space and time. So today I'm going to focus on this first question about convergence, and I'm going to use geophytic plants, which I'll define shortly, as a really good example of convergent evolution. Geophytes are herbaceous plants that resprout from below ground buds. And if you want to know more about geophytes, feel free to check out this review paper that my collaborators and I put together on some of the evolution, morphology, and development of geophytic plants. Geophytes have evolved multiple times across the vascular plant tree of life. So geophytes are not a lineage, it's a growth form that has many different independent evolutionary origins, including in diverse groups like ferns, lycophytes, and various groups of flowering plants. Geophytes are not only diverse in their evolutionary origins, but they're also diverse in the types of underground storage organs that they have. So most geophytes have some type of underground organ that allows them to store things like starch and water below ground. And those reserves then allow them to fuel this regrowth from their below ground buds. And the types of tissue that is modified to produce these below ground organs include sort of the range of general plant tissue types that exist, from leaves, stems, and roots. So modified leaf tissue can produce a storage organ known as a bulb. Great example of a bulb is an onion. And the layers of an onion are actually sheathing leaf bases that have been modified for storage. There's also modified stems that are underground storage organs. A couple of examples of those are corms. My favorite corm is Calo or taro, and that's a sort of really important tropical food crop. And then rhizomes as well, which are these horizontally elongated underground stems. A good example of that is ginger. Some people like to say ginger root, which drives me a little mad. It's actually a stem. Yeah, I'm not really a good person to go grocery shopping with. But plants also do modify root tissue for storage below ground as well. And these modifications can take different forms. And so here I'm just sort of roughly grouping them into elongate and rotund root tubers or tuberous roots. I'll use those terms interchangeably. So these are sort of some of the main examples of different types of underground storage organs that geophytic plants can have. We generally think that geophytic plants are adapted to seasonal climates. And a couple of examples of that might be a really good example are Mediterranean climates, which are characterized by cool, wet winters and hot, dry summers. And geophytes are known for being particularly diverse in Mediterranean climates, where they're able to regrow in moister conditions following the seasonal drought. But they're also common in the understories of deciduous woodlands, where geophytes are able to quickly sprout and flower prior to the trees flushing out and blocking light to the canopy floor below. And this is a picture that I took just a couple of days ago when it was sunny out here. And we can actually test this or quantify this seasonality. And so for the rest of the talk, I'm going to be describing two different aspects of seasonality. One as the seasonality of temperature, and the other is a seasonality of precipitation. And this is taking sort of a global view of these climatic variables from WorldClim climate database. So I wanted to ask if the repeated evolution of the geophytic habit is due to convergent adaptation to seasonal climates. If yes, then despite these morphological and developmental differences in their below ground organs, geophytes are adapted to similar climates. Alternatively, the morphological differences between these below ground organs, whether it's leaf, stem, or root tissue, may mediate different organismal responses to the environment. And I decided to test this in the plant order Liliales. Liliales has around 1,200 species in it, which was roughly the size of the data set that I was looking for this kind of study. But most importantly, it includes representatives of all of these different types of underground storage organs that I told you about. So it's a really great group to examine the role of morphological differences in underground storage organs. But it's not a great group for testing like geophytes versus not geophytes because most Liliales are geophytes. In order to test this question, is the repeated evolution of the geophytic habit due to convergent adaptation to seasonal climates? I needed to overcome three major challenges in phylogenetic comparative methods. The first is that we often think of phylogenetic comparative methods as thinking about traits or characters and their character states. Often, character states some states are going to be more similar to each other than others. And so I wanted a way to accommodate that hierarchy or different sets of relationships, nested relationships within my states, particularly in the case of geophytes, where we know that some of these underground storage organs are modifications of similar tissue types. And so we might expect that, for example, a rhizome is going to be more similar to a corm than either of them are to a bulb, because the bulb is modified leaf tissue as opposed to stem tissue. And so I needed a way to bring in sort of these relationships between the character states into my model. You might think of sort of this hierarchical structure as being rather similar to an ontogeny or an ontology. And we think often ontologies are used in gene ontologies and thinking about sort of the role of molecular processes. My collaborators and I are really trying to advocate for the use of ontologies in plant morphological and evo-devo studies, because we think it can facilitate these types of analyses that take into account the complexity of nested relationships between character states. So it turns out that there's been some recent developments in structured hidden Markov models that allow you to take into account these set of hierarchical or nested relationships between states. I'm not going to go into the details of the structured hidden Markov model, but feel free to ask me a question about that after if you're interested. I'm really excited about this approach. It's the Paramo pipeline by Sergei Tarasov. It's not something that I developed. So this was sort of the first, right? We have this problem of hierarchical relationships between our discrete states in our model, and we can use structured hidden Markov models to accommodate those relationships. The second issue is the longstanding challenge of correlating discrete and continuous traits in a phylogenetic framework. There's been some really exciting recent development in this that's sort of occurring in parallel to this project, including by James Boyko, who's a postdoc here. So there's been sort of really exciting work on developing multiple approaches to this problem. And I just told you about the discrete trait in our model, which is these types of different underground storage organs, but we also have this continuous trait, which are these climate variables. And before I go on to sort of how we dealt with this, I just wanted to dive in a little bit into sort of where this data came from in our case. So this was work done by an undergraduate student who's now a master's student at the Universidad de Chile in Santiago. And Abby developed a pipeline in R to facilitate large scale climatic niche modeling across hundreds of taxa at a time using MaxN. And so essentially her pipeline takes in as input the species that you're interested in working with, and it downloads and facilitates automatic cleaning of occurrence data, and then running these models and producing sort of a cleaned and neat output. So you can check that out on the GitHub page there if you're interested. Okay, so back to our issue of dealing with discrete and continuous traits. So we have these climate data, and for the discrete trait, we use the structured hidden Markov models. And then what we did was separately model the evolution of the continuous trait using what are called multi-optimal OU models. Again, I'm throwing these terms out in case you're interested, but don't worry if you're not totally familiar with these models. That's not super necessary for understanding the main point of the project. But feel free to ask me questions after this if you want to know more. The most important thing to note here is that we think that multi-optimal OU models do a good job of capturing the amount of variation that we think is present in climatic data, and do a decent job of modeling the evolution of that kind of trait on a phylogeny. So when we run our structured hidden Markov models and our multi-optimal OU models on our discrete and continuous traits, respectively, we end up with something that looks like this on the slide. So we have here two phylogenies that have different colors on the branches. The different colors correspond to different character states. So we're going to take a simplified example here, where on your left, the phylogeny in green has darker green, meaning that those lineages have a bulb. And in the lighter green, it means that those phylogenies do not have a bulb. On the other side, in this blue tree, we can think about these colors as representing regimes of climatic evolution, where the optimal value of climate in for the regions of the tree in light blue is going to be less seasonal than the regions of the tree in dark blue. So these are the results of running those two models that I told you about for each, the continuous trait and the discrete trait independently. What we can then do is ask to what extent those evolutionary histories are similar. So imagine lining these two phylogenies on top of each other. If we see perfect alignment where every time one history changes from light green to dark green, the other tree changes from light blue to dark blue, then that indicates perfectly correlated evolution, because every time one thing changes, the other thing changes too. So essentially what we're doing is quantifying that degree of overlap in the evolutionary histories of these two traits. And so we do that by estimating what I call a state averaged value. So for each state of the discrete trait, bulb or no bulb, we estimate a range of seasonality of precipitation values. And then we can ask whether these two distributions are statistically different from each other. And this was essentially our solution to how you can accommodate or test for an association between discrete and continuous trait evolution, leveraging the power of stochastic mapping. Okay, so the third issue is this question of imperfect correspondence between traits. So imagine that we have here a climatic niche. We think of climatic niche as being an inherently complex multidimensional object that's inherently influenced by many factors beyond underground storage organs. There's no way that I would ever think that underground storage organs are the only thing contributing to the climatic niche of a group of plants. And so what we want to do is ask whether there is a statistically significant association between this morphological trait of interest, underground storage organs, and the climatic niche, while taking into account that we expect there to be other factors that are contributing to the variation of that continuous trait. And so the way that we dealt with that was in part the way that we developed the method of lining up those stochastic maps allows the continuous trait to evolve in a way that can accommodate a lot of variation. But we also then compared our empirical data to a realistic null model based on simulations. And again, I'm not going to go into details about that procedure, but I'm more than happy to answer questions after this. So to sum all that up, we developed a new method or pipeline to overcome major challenges in phylogenetic comparative methods. And we did this by leveraging stochastic mapping. That's the act of painting those colors on the branches of a tree for the discrete and the continuous trait, and then quantifying the degree of overlap between those two histories. So I'm going to give you a preview of what the results might look like. So on the x-axis here, we have our state averaged values. And then this is going to be density plots. So you can think of the y-axis as density or frequency. And I'm going to show you this for the state averaged value of the seasonality of precipitation and the leaf character, which is looking at bulb versus no bulb. So these are the results. We have our light green, which is not having a bulb, having less seasonality, lower values than the dark green having a bulb. And so this, to me, I got this result. I was like, great, makes sense. You have a bulb, you tend to grow in more seasonal climates. However, when we compared these distributions to what we expected under our null model, it was completely non-significant. This fit entirely under a model of random evolution where there was no association whatsoever. So this difference between these distributions is not significant. I did this procedure for 8 different comparisons. So for our 2 different climatic variables, seasonality of temperature, seasonality of precipitation, and for 4 different morphological characters. One where we just looked at whether or not leaves were modified, one where we just looked at whether or not stems were modified, whether or not roots were modified, and then finally the combined holistic phenotype, thinking about modifications to all of those 3 different types of plant tissue. And what we found is that the only significant comparison was for the root character and seasonality of temperature. So I'm going to show you that plot here. So what we found is that plants that have root modifications in the form of root tubers occur in more temperature seasonal climates than plants without those modifications. Or sorry, less, the opposite of what I just said, less seasonal climates. And so this is really neat because this implies to me that the variation in degree of seasonality is linked to the type of tissue that's being modified in producing these organs. It's only when we think about whether that organ is a root or not a root that we see this difference in the correlation with these climatic variables. And so this also suggests that convergence in the geophytic habit is probably not being driven by temperature seasonality, potentially by precipitation seasonality as well, or other climatic variables that we didn't test. This also, I think, is a really good example of the importance of integrating morphology, plant development, and novel statistical methods to produce new insights into the nature of convergence. And I also think that this method that we developed is a really exciting way forward in phylogenetic comparative methods. It's super flexible for different sets of empirical questions. So feel free to check out that paper. It's available now in systematic biology. So in the time that I have left, I want to tell you a little bit about what I'm currently working on, which is interested in a similar set of questions about convergence. When we see convergent at the macromorphological level, do we also see it at the anatomical level and at the genetic level? And I'm testing this sort of set of questions using my favorite group of plants, the genus Bommeria. These are tropical plants in the order Liliales, so they have rhizomes and root tubers, and they have these really charismatic flowers. But most important for this study, they also have variation in growth habit. There are some plants that most of the species are climbing, but there are some plants that have lost the climbing habit and are instead erect herbs. And we think that this change has happened around 4 times in the genus, if not more. And I think one thing that's really interesting is that these erect plants tend to grow in higher elevation environments. And so I'm also interested in this question about how do these adaptations to extreme environments evolve over a macroevolutionary timeframe. And so this is, I think, a good example of that. So, the main goal here is to ask whether repeatedly evolved phenotypes are the result of the same molecular and anatomical mechanisms. And so to do that, I've been collecting data on stem anatomy, on the sort of the genomic level data of these plants, and then I'm going to put that into a macroevolutionary framework. The end goal is to develop a phylogenetic comparative method that will directly test for specific nucleotide changes that are associated with phenotypic change, and then be able to test this across a phylogeny. I think what's really exciting about this is that maybe we'll find that there are some molecular changes that are universal across the entire phylogeny, but perhaps there are changes that are clade specific, and we can start teasing that out by looking at this in a phylogenetic framework. So with that, I want to thank my co-authors on this project and my funding sources, and all of you for listening.
This might be a really naive question, but do you think your lack of strong associations between seasonality and storage organs in the first part is just due to functional redundancy? In other words, does it matter to the plant if it's a bulb or a rhizome?
Right.
And since you're doing it pairwise, is that why you?
Yeah, so that's a great question, and I think that's one of the main things that I wanted to test. So I think the previous sort of hypothesis in the literature were that there was functional redundancy, but it doesn't matter if you're a corm or a bulb. You look at the outside of those corms and bulbs and they look the same, right? Like they seem to be serving very similar functions for the organism. And what we found was that there appears to be functional redundancy, but not with the root structures. And we don't totally know why that is. I suspect that it might have to do with the particulars of this plant group, actually. So the liliales that have tuberous roots most often also have rhizomes. And so it's possible that in those organisms, they're actually partitioning the sort of storage roles into the root structures. And the rhizomes then also serve a clonal or sort of like, clonal reproduction or spread function. And so it's actually what we're teasing out is like that partitioning rather than the root structure itself. But I'm not totally sure what's going on and like why the root derived structures were showing up as being the ones that were different. But absolutely, I think there's functional redundancy. It's just kind of weird to me that you see such functional redundancy within one relatively small evolutionary lineage. This isn't saying like in liliales you have stem tubers and then that's functionally redundant with the bulbs of oxalis or something like that. So I'm not, I think there's still sort of an interesting question about why do we see so much morphological diversity in these structures when they appear to be mostly functionally redundant.
Questions? Okay, well, let's say Carrie.
Yeah.
Where's the break?
So
Here, share screen. Screen two. Right there, and finally getting it. With cursor. Floating meeting controls.
There we go. Well, it doesn't look much different, but that works.
That's fine. We're good, right? And this one functions, we think.
Okay.
Cody is currently a postdoctoral scholar in the Department of Ecosystem Science and Management at Columbia's College of Agricultural Health Sciences. Currently works with Jill Hamilton. Cody received her Bachelor's of Science in Biology at Virginia Commonwealth University in 2004 and her master's teaching in 2005. Strength of a non-traditional academic journey. But she returned for a Master's of Science in Ecology and Evolutionary Biology to BCU in 2017 and completed a PhD in integrative life sciences last year, 2022. Having now expanded upon graduate research in evolutionary genomics, studying reproductive isolation and adaptive introduction in populace, one of my favorites, which we'll hear more about today. So please join me in welcoming Connie.
Yes, thank you. I'm Connie the Optimist, clearly by this title. I have hope in hybridization. I'm interested in how hybridization influences divergence and also adaptation. But first, I'll take you to this hopeful landscape. We've got these beautiful green, luscious foliage hanging on to these trees, which gives us an indication that these organisms have traits that confer survival within this particular space. Of course, to have hope, we have concern first. From that is which it is birthed. And unfortunately, our landscapes are transforming to look more like this. These are evergreen trees that are not green. They don't have leaves on them. And this is really becoming more and more apparent that trees are having a hard time migrating and adapting to our changing climate. And that's because of their long generation time. Sally Aiken and her colleagues, they wanted to quantify exactly mechanistically how effective migration versus adaptation, or the two of them together, were moving into the future. And I changed these axes for better interpretation, although I made a mistake. It should just be genetic diversity. Just cross out the lost there. And time into the future. And so you can see that moving into the future, and this last tick on the x-axis is like year 2100. We see that we have genetic diversity kind of lost in our tree populations. Even if we consider migration and adaptation together, but that's the best case scenario. What really I want to focus on is the fact that when you look at migration and adaptation separately, adaptation's the best bet. And in order to have adaptation, we need genetic diversity, and gene flow is a way to get genetic diversity quickly. You can have intraspecific gene flow, which is within populations of the species, or you can have interspecific gene flow. I'm focusing on introgression, which is interspecifically across species boundaries. And that happens through hybrids, through this mechanism called backcrossing. What's awesome is that it produces novel genetic recombinants. These can have major fitness effects within certain environments, and these are often linked to hybrid zones. So, justifying hope, once again. Introgression increases genetic diversity, which increases adaptive potential. And we're finding in these speciation studies that the divergence histories of these forest trees are often 99.9% of the time characterized with a history of introgression. So myself and some of my colleagues are really starting to think that introgression may be an evolutionary mechanism that these trees rely on for genetic diversity. And this would help counteract the long-lived, sessile, long generation time characteristics under a rapidly changing climate. I did a Google Scholar search because my PI was like, let's write a review about hybridization in forest trees. And I was like, let me see if it's been done. And you can see there's been a lot of interest using these three keywords here. And that really the onset came with the increase in genomic resources. And with those genomic resources, we found out that there's a variety of outcomes with hybridization. And if you look at these 4 consequences from these 4 different outcomes, 3 of those 4 have gains, right, to biodiversity or genetic diversity. If we add a 4th term, introgression, you can see there the relative contribution of introgression in the literature. I hope to be one of those one day. I'll be an extra little tick mark. This paper, though, did not focus on forest trees, but I found it really inspirational to my work. Hybridization and hybrid speciation under global change, and super relevant for the theme of this symposium. In this review, they talked about hybridization with non-native species. Really big concern right now. It's not all hopeful, right? Hybridization. That we're seeing an increase because of a breakdown of geographical isolation. And some of that is anthropogenic dispersal. And some of that's natural distributional range expansions with a warming climate. And they also talked about some experiments about that observed a breakdown of prezygotic reproductive isolation, and that they're seeing shifting reproductive phenological schedules, such as timing of pollen release. And this really, when Meredith was giving her talk, I was like, oh yeah. I decided to give you guys a little schematic of how this would work for forest populations, even though that this has all been done on short generation time plants. So no hybridization would be expected between these two species because their pollen release dates are diverged. And the idea is that of those two species, one maintains their pollen release date, the other one has a plastic response to a warming climate, such that eventually they overlap and we have potential for hybridization. Quantifying this in forest populations is difficult. So what we can do is really think about, again, how prezygotic isolation might break down cyclically over time. Consider the fact that are these periods of glacial times when we might have contractions and isolation actually long enough for reproductive isolation to form? Most of these divergence histories for forest trees go well past this x-axis, like into 1 million to 15 million years ago. So they have experienced a lot of this variation. And word at these interglacial periods, I don't know where this little thing is. Is it? Oh, there it is. All right, the interglacial period was that gene flow is kind of the question. So we can take DNA, we can look at it and infer things from the past. You can see here my friend's dog, it's past. We can do this for tree populations too to see how they've responded to global changes. And we use demographic inference for that to elucidate these historical events. We can look at effective population sizes, which would be here kind of denoted in U. Divergence with gene flow would have an arrow between the two lineages or without strict isolation. Rate and direction of gene flow, you can do kind of an asymmetric to see if it's unidirectional. And then the timing events can even be parameterized as well. So this first case study I'm going to show is from my dissertation work. It's 2 pine species. They have gigantic genomes. So we use RAD-seq data, which is basically taking samples of their genome. So genome-wide representation of the variation between these two species. And they're native to the Appalachian Mountains, which is kind of a characteristic spot for phylogeographic breaks and lineage divergence. This project actually started with assessing the diversity of table mountain pine because they're high elevational species with fragmented populations. We're like, genetic diversity is probably small or they're at risk. And we had fire suppression practices over this past century, which for this fire dependent species kind of accelerated the decline. So this hasn't gone unnoticed. NC State and the USDA have banked their seeds. There's management practices doing prescribed fire, but no one had done a genetic assessment on it yet. Like, and then while I'm at it, I'm going to assess hybridization with like its close relative, pitch pine. You can see from growth form, they're really similar. They've got this really cute bonsai thing going on. When you get up close though, they definitely have striking differences in morphology and like needle length and cone structure. But they do have overlapping distributions, which is interesting. And these sympatric stands are the only stands that have been studied historically. And the last time they were studied was in the 1960s. Hybridization was deemed rare within these sympatric stands, and pollen release date was separated by about 3 to 4 weeks. So I did range-wide sampling of these 2 species. That was fun. Best shape of my life, climbing those mountains. And performed admixture analysis as the first indication of whether hybridization has or is happening. In this analysis, these are basically 126 samples. Each sample is a vertical bar. And if it has both blue and orange, then it has mixed ancestry. So a sign of introgression there. And you can see that this introgression or admixture is present range-wide. For this PCA, this kind of gives us a first indication of the amount of genetic diversity that's there. I was expecting way less space to be occupied by the Table Mountain pine samples, given their fragmented populations and small distribution. So it's kind of an open-ended question, but maybe this admixture here is adding to genetic diversity. Testing demographic scenarios, believe it or not, this is a subset. There were like 100, 1,000 different choices. But this is what we landed on. And in this demographic inference, what we're doing is we're taking patterns in the joint site frequency spectrum data and trying to find a model that best matches those data patterns. And the best fit scenario, given my fragments of genome data, was one that allowed population size changes and migration change over 2 time periods. And when we look at these, we can really see how climate influenced the divergence of these 2 pines. The onset of divergence aligns with the onset of the Quaternary period, which is linked with adaptations to seasonality. And we see a major population size reduction that happens at the last glacial maxima or last glacial period. And we see that their history has had introgression happening throughout. This model is a simplification, certainly, of their divergence history, but it's what could be captured in the genetic data. So that makes us then understand, like, well, okay, there's been gene flow throughout divergence, but they're in sympatry. How are they maintaining their species boundaries? And so we look to some redundancy analysis, of course. In this RDA, we looked at climate variables and geographic variables to see what was really separating these 2 species, and elevation and bio-15, which is precipitation seasonality, were the 2 main drivers of genetic structure differences between the 2. While I'm typing up this manuscript, this like macro scale pattern study is published from Jen et al. And I was like, what? No way. 2 of 5 drivers of species diversity in pines of this region, Eastern North America, were elevation and seasonality. Like, that's cool congruence. And then we have the pollen release timing separation as a major thing. We've got seasonal climate also involved in their divergence. So pretty amazing what you can glean from 2,000 genome-wide SNPs. Can you imagine what we could do with 20 million SNPs and a known hybrid zone? That brings me to my current postdoc work. So 2 popular species, very different life history traits than these previous 2 pine species I studied. They've got compact genomes, which means we can affordably sequence them in our model system, and they're riparian species of the Rocky Mountains. And just to give you a quick comparison of the landscape we're dealing with here, we had our pines over here, and we've got the well-named Rocky Mountains on the West Coast. We'll put that into, that'll be helpful later as I tell the story. So we've got the hybrid zone of these 2 popular species. Where their distributions begin and end are the Rocky Mountains, but there is a really narrow hybrid zone. These hybrid zones are natural laboratories for studying the development of reproductive isolation, patterns associated with introgression. And from Suarez-Gonzalez's work, we already had an indication that there was introgression happening. We have evidence of balsamifera being introgressed into trichocarpa, but it's not genome-wide. There's some at chromosome 15, not much at 12, some at chromosome 6. And these are the only 3 chromosomes that she had analyzed. And with phenotype data, these were related to like osmotic stress and cold tolerance. Like, oh, adaptive. when I say we, it's not really me. It's these 4 PIs who spearheaded this. And they came together, and the major goal is to explore and describe the genomic architecture of forest species boundaries and how that relates to climate and hybrid breeding. They did the transect sampling, which is an amazing design to capture the parents and the hybrids across the zone. They hired me to do the genetics. And so here you see some really cool patterns across a principal component space, where you can see that when you compare our ancestry coefficients from admixture analysis, we've captured a lot of trichocarpa over here, one species, and a lot of genetic diversity within it. Balsamifera here, and a gradient of hybrids in between. But we see that these hybrids definitely have some parental influence, right? Where they are and who formed them determines their genetic position in this PC space. When we flip it to look at PC2 and PC3, we start to see some other structure surface. And whoa, one back. These are the northern populations, Alaska and Cassiar over here, are really segregating from the middle transects. So I anticipated to have a K equals 3 as the best fit cluster value based off of those PC patterns. And indeed it was. What I didn't expect was that the FST value was going to be similar across all 3 populations. And this definitely has a story to tell. So these are pairwise comparisons. That means that northern trichocarpa is as diverged from southern trichocarpa as it is from the other species, Balsamifera. Again, the end goal is to determine which of this is adaptive. So we have to tease apart the neutral and non-neutral processes. And that's a challenge because these interact on so many levels, but I'm going to show you my efforts. It started with the EEMS. That is where we can identify barriers to gene flow. And what this does here is the white is basically the expectation based off of isolation by distance. And any deviation from that is a deviation from IBD. So if it's brown, then we have low gene flow or a potential barrier to migration. If it's blue, they're connected. So 2 things really pop out here. We've got apparent isolation in our northern transects. So we've got some genetic differentiation that's happening there, probably because of landscape barriers. And then we also have the Rocky Mountains, which are helping separate and maintain the species level boundaries between tricho and balsam. Now trying to look at non-neutral drivers, a couple of takeaways. We've got continentality as the main driver of differentiation between, or associated variable of differentiation between these 2 species, temperature diurnal. With higher temperatures associated with trichocarpa, generally speaking. But then we've got that structure within trichocarpa we've got to consider when we're looking for adaptive integration, to have the drier populations in the south and the more precipitous, the associated populations in the north. We also have just being north as a strong thing for these northern populations. So next thing, again, trying to tease apart the neutral and non-neutral processes, we look to chloroplasts, which you can actually get from your whole genome sequencing data. Michelle Zavala is the PhD student that's also on this project and that I mentor, and that she mentors the undergrad, Brianna. So we've got a little tic-tac-toe here. And the divergence history without gene flow can be kind of discerned from these patterns, as well as migration and patterns of local adaptation. From their analysis, they found 3 clusters. And so I'm going to compare the nuclear and chloroplast for you all side by side, but I'm going to change everything in that top cluster to blue. All right, quite comparative. But I want to zoom in real quick here on Chilcotin, this one transect. We have a potential case of chloroplast capture. And there's a lot of studies out there that talks about cytonuclear incompatibilities, but some are surfacing as offering some real fitness advantages. And so we are going to have to look deeply at some of the phenotype data that we're gathering on the fitness consequences of this tricho identity in nuclear, but balsam identity in chloroplast. I was curious because we had the FST estimates from the nuclear data that was equal across the pairwise comparisons, what the divergence history would be from these chloroplasts through pairwise comparisons. And so what I saw was, given the confidence intervals here, we have a decently deep divergence between 3 and 4 million years ago. And when you look at that with the confidence intervals, they're about equally diverged. So this landscape has had a lot to do with the differentiation, I imagine, as well as these oscillations over time. So we definitely have our hands full trying to pick out the adaptive things, and then also trying to look at the divergence history from nuclear data, given that we have 3 structures here. So this is currently under construction. I was hoping to have that for you, but I don't want to present anything that I'm not confident in quite yet. What we can say is that this hybrid zone has varying degrees of ancestry. We've got climate and geography explaining some of it, barriers to gene flow, and signatures of selection and introgression may differ by transect. They are going to differ by transect. So we're going to look at them independently. Moving forward on this project, we've got a lot to look forward to, especially since we're going to have the phenotype data, which is really going to tell us about adaptive introgression across these 50 common gardens in the US. But the big picture here is hybridization during the history of divergence is a growing expectation for forest trees. Natural hybrid zones are valuable. Introgression may be a mechanism that these trees employ to gain genetic diversity. But we should still be concerned because, like some other presenters today have mentioned, we've got anthropogenic land use that may be or have already disrupted gene exchange. Thanks.
Thank you. I'm just wondering, why do you think that these species maintain their species identities through hybridization? Do you think ultimately they will just sort of disintegrate and become a single taxon, or is there enough genetic diversity in the planet together that they're only allowing for a small level of integration?
Yeah, I think, well, genomically, it's an interesting question because I think it matters on the 2 species, their history before coming into a hybrid situation. Are you talking about poplar or just in general? Poplar? Yeah, so if the introgression, these 2 poplars, they have extensive linkage disequilibrium across their genome. So there's a lot of any introgress variation that gets in there is going to get purged really quickly or get selected all the way to adaptive status, right? So I think it's a matter of what's happening in the environment when that introgression is happening and how the genome decides whether it's fit or unfit within that environment. But I think that a lot of what is purged is environmentally based because of that maritime continental difference. So it comes in and then Trichocarpa is like, no, balsam, I don't need you. But if the environment's changing and it introgresses and balsam's like, wait, maybe I do need you. Maybe right now is a good time to keep you in my genome. It's a way to think of it.
More questions?
Thanks for that talk. I have a sort of more mundane, like methodological question, which is like, how was it for you to do DB-RAD with such large genomes? I work with like pretty large genome plants, and I've always been a little concerned about DB-RAD because just you might end up with like a lot of discrepancy between the sites that are cut in one individual versus another if you're working with like such a huge genome. So did you worry about that at all, or did you find that it was like a manageable amount of missing data?
There was, we pruned a lot of missing data out. So no, there was a lot of missing data from RADseq. I think we started with like 81,000 SNPs and ended up doing that analysis on 2,200. So yeah, there was a lot of that. In another study we did use angst to impute, like to deal with the missing data. That was a good question. It's absolutely a thing. Yeah, but that demographic inference, like it absolutely has had more complex history than what we could capture with 2000. But I think it's still pretty cool that we can glean what we can, yeah, from a cost-effective method. Yeah.
We have one more for this one. March.
So, in this next phase, when you're talking about incorporating phenotypic information, like what are the key things while I bring to that incorporate information?
Yeah, good question. So we have a bunch of ecophysiological traits, like stomata conductance and stomata density. Given that there's a lot of those precipitation related variables across trigo, I think that will be important, especially with drought coming about. And then we have, of course, the typical height, SLA, yeah, a whole team of all these mini gardens are at universities, and they've got a team of like researchers who are doing, I think it's like 25 different phenotypes. So, yeah.
Wonderful. One more round of applause for Connie.
Thank you. Yes.
And I'll send people the
When you're ready.
I think we're getting there. Screen 2. There. One. Hide floating controls. Blow it up full. I think we're good. Yes.
Thank you.
Hi, everyone. I have the pleasure of introducing Dr. Susanna Vollmeyer, who is from Davidson College. And I was excited to nominate her as a speaker, one of our keynote speakers, especially because she has an active research program on climate change and is at a small liberal arts college. So if there are any students here that are interested in thinking about trying to get a job at a SLAC, and as well, she has done an amazing amount of DEI advocacy in the Society for Study of Evolution. So please feel free to talk to her about that as well. So let's welcome Susanna.
It is, I feel very loud right now. It is a pleasure to be here. This is my first in-person talk since, before COVID was a thing. So I'm very excited to hear some really good science and chat with you all. I thought I would take the opportunity today to share this sort of thread through my research over the years that led to a grant that was funded and that we're starting this year. So I'm going to start with my graduate work through postdoc work and leading you to like what a lot of my focus will be over the next couple of years. Let's see. Oh, it doesn't. I can do this. Okay, so a tiny bit about me. I'm an assistant professor at Davidson College. I've lived in many places, as you can see there. I'm a plant evolutionary ecologist, and I have 2 young troublesome rugrats at home. They found dry erase markers and surprised me. And the topic of this symposium couldn't be more timely, given that the IPCC, the Intergovernmental Panel on Climate Change, just released their latest synthesis report within the last week. And this is a figure from that report. I'm not going to walk through it because I want you to go look at it and read the report. But we now have very strong evidence that with every increment of global warming, regional changes in mean climate and extremes become more widespread and pronounced. And we're now beyond the point where these reports are even including a scenario of 0 degrees warming. We're past that. Now we're hoping for 1.5 degree warming globally as our optimistic target, right? And then of course we can see increasing severity with warming from there. We also know from climate change that's occurred so far that it can impact evolutionary and ecological processes at multiple biological levels of organization. This is a really neat paper that it's a sort of meta-analysis that goes through climate change studies that have shown impacts at these different levels. But we have evidence that climate change is impacting the genetics of populations. At the organismal level, we see physiological and morphological effects of climate change, also phenological. We've seen that come up a couple of times today. At the population level, we see changes in population dynamics, their distributions over space. And then at the community level, we're seeing changing interactions between species. And even at the ecosystem level, productivity can be changing for a variety of species. So the effects of climate change are widespread. They impact everything from genes all the way to, ecosystem function and ecosystem processes. Within populations, we often see, within species, we often see populations harbor many characteristics that may make them prone to respond to climate change in different ways. So if you can imagine a hypothetical species distribution over there on the left, maybe there's a continuous portion of the range in the middle. You've got a leading edge of the range where many species have shown to be shifting their ranges in response to climate change, perhaps higher elevations or higher latitudes at the leading edge there. And then a rear edge where we've actually seen many populations at the southern or low elevation end of these distributions are going locally extinct, and we're seeing a contraction of the range at the lower elevation or lower latitude range of many species. And what's interesting about species distributions over space and then of course through time is that they differ in a whole bunch of their dynamics and genetic structure in this way. So to save time, I'm actually going to go through this, but this paper walks through how factors like population age or even within population diversity can differ across the range of a species and impact individual population responses to climate change. I became very interested early on in these dynamics, but also what conservation practitioners can do with this information, right? When I started my graduate program or my graduate studies, there were a lot of debates in the literature. I'm talking one of those back and forth, like replies and responses back and forth about some of these, what seemed like really drastic conservation measures. One of them was assisted migration and the other was assisted gene flow, just to kind of explain what those are. I don't think I have a laser pointer. Actually works. Okay. So assisted gene flow is a conservation program that's proposed mostly for saving threatened populations from within a species range. So if you have a threatened population, generally you're thinking it's got lower genetic diversity, it's unable to respond to selection and adapt in time with whatever pressures that it's experiencing. But if you could bring some genetic material from a different population in the range, one that may be exposed already to some of those stressors, you could introduce some adaptive genetic variation into that threatened population. Ideally, they would hybridize in some sort of way, and you might get some evolutionary rescue occur. So that's the assisted gene flow program. And we've done this already with a couple of species. There's some really, from mammals all the way to plants, there's some really good success stories with this strategy. The more contentious one is trying to anticipate the effects of climate change on species and where they will be distributed in the future, and perhaps trying to help them migrate to those future suitable habitats ahead of time. And that's called assisted migration. It's been named like 5 different things in the literature. And ideally, you would, do your due diligence, find some climatically suitable habitat outside of the range, but in the direction that migration would be taking that species naturally. And you'd preemptively move individuals to that new spot beyond the range. And you should go back and look at the literature because they were really arguing, you're going to bring elephants to the Midwest and you're going to do, it was really contentious at the time. And the number of commentaries and reviews on the topic were skyrocketing, but there were no papers that were just like asking how feasible it is or what are the basic limitations of programs like this in practice. So that's where I started out with my graduate studies, being interested in that topic. So here's what we'll talk about today. The effects of climate change on a couple of things. We'll start with the effects on assisted migration and assisted gene flow programs beyond the range. Then we'll move to postdoc work I did looking at the effects of climate change on local adaptation within the range. And then I want to end by describing to you a collaborative grant that we just got funded that mixes that all together and looks at the effects of climate change on within the range and beyond the range, ecoevolutionary processes and population persistence. So let's start with these conservation management plans in Chamaecrista fasciculata, which you've seen in a previous talk today. One reason I'm so interested in these programs is the fact that we talk about translocating populations across latitudes, for example, but there's so many environmental variables that we know vary across latitudes in really predictable ways. So for example, on the left, you can see latitudinal variation in temperature over the year. And on the right, you see latitudinal variation in day length or photoperiod over the year. And a lot of organisms, and especially plants, rely on specific environmental cues as signals to proceed to the next developmental stage. And sometimes they need combinations of those specific cues or even like sequences of those specific cues in order to proceed developmentally. And so with climate change, we know temperatures are increasing already and will continue to increase. But one thing that will not change is photoperiod, right? That is set for a given latitude. So whether you stay put and you don't migrate, and you experience increases in temperatures, or whether you migrate and thus experience different photoperiods, those two particular environmental variables are going to be decoupled for you, no matter what happens, right? And so I became really interested in that particular angle and still am of these relocations and also what it means for species that are migrating across latitudes. So keep this in mind as we proceed through the discussion. So will factors like this actually impede assisted migration and assisted gene flow programs? So this is Chamaecrista fasciculata. It's an annual legume. It's really pretty. You can even find it around here during the summer. It grows and flowers indeterminately until first frost. And I have some evidence that I'm not showing you now that flowering time is photoperiodically determined in this species. So that can be important. This map, all the dots here are showing you, this was a map by my collaborator, Seema Chef, and all these dots are showing you where the distribution of Chamaecrista is found in the US. So it's all over the US with really kind of abrupt range boundaries at the Canadian border almost. And then in the Midwest through the US, right? It's a very interesting distribution. And what I did as a graduate student, I'd never seen this plant. No one I knew had worked on this plant, but I drove all around and tried to find it. I collected seeds from a bunch of populations, and I'm going to focus on 4 for this study, which are shown in the red stars. So 2 northern, Minnesota and Pennsylvania, and 2, I know I'm going to call them southern, but they're really mid-range, right? Missouri and North Carolina. And what I did was collect seeds and then ultimately use them as like candidate populations in assisted migration programs beyond the range to my field site in the yellow triangle there. In Southern Ontario. That's the Koffler Scientific Reserve, which is associated by the University of Toronto. At the Koffler Scientific Reserve, we installed experimental climate warming arrays. So you've seen these twice now today, which is great. This was when they were being installed. They're steel triangular structures that are anchored into the ground. And for each one, you've got 6 infrared heaters that are like angled down from the horizontal. And they create a uniform heat shadow within that really big plot that allows you to apply a warming treatment in a field setting, even to larger plants. And so what we can do is set those heaters to raise the temperature within the plots to some desired degree above whatever's being registered in the control unheated plots. So we aim for a modest 1.5 degree daytime increase and 3 degree nighttime increase for this study. So all 4 populations planted in heated and unheated plots in the field beyond the range. And I wanted to start by showing you some phenology data. So on the x-axis are my 4 populations arrayed from northernmost on the left to southernmost on the right. A stands for ambient and H stands for heated. And we'll go through 3 different traits here on this graph. First, I monitored when the plants produced their first flower bud, that signal to go reproductive, right? When did that happen? And here's what that data looks like, budding onset. We have a latitudinal decline in reproduction, which is not surprising and super common in plants. The northernmost plants start reproduction earlier than the southernmost plants. And across these populations, in the heated treatment, warming advanced the onset of reproduction, right? And did so consistently. So there's no variation in that plasticity across those populations, those reaction norms. The slope of those reaction norms are pretty parallel. I did the same for first open flower, which is the trait that's most commonly monitored, sort of the poster child trait for climate change. And we see exactly similar patterns, right? Earlier flowering in the north, later flowering for the southern populations, an advance of flowering in the heated treatment that's exactly proportional to when the flower buds were produced, right? I also monitored the date of first mature fruit. I think this is a hugely consequential trait, given that that's when the offspring are put out into the world. But we don't measure it very often because it can happen so much later, and it's hard to figure out what you call the date that a fruit is mature. So in this plant, I called that date when the pod had turned brown and started to rattle. So this species actually the fruit explode dehisively, and it'll fling its seeds everywhere. It's like hearing your fitness measures be lost in space when it happens. It's like a little heartbreaking. But anyway, so that was my measure of day of first mature fruit. And then I'm showing you that data at the top of this graph. So you can see now why the y-axis is so long. This is like the longest lived annual. It just keeps going until first frost. This trait ended up being the most interesting because we see first off, the biggest advances, like the biggest responses to warming were in the day of first fruit. But also we see variation in plasticity across these populations, such that, except for the southernmost population, fruiting advances more the further south you go, or the further south those populations are from. That southernmost population, those poor things, it just, very few individuals actually made fruit. So it looks like no difference, but I just have a very small number of plants actually contributing to data from that population. They're just so long-lived. Here's what's going on with phenology beyond the range. And again, remember, I have evidence that phenology is like limited, or not limited per se, but governed by photoperiod. I wanted to look at plant size next, and here are the plant sizes for these plants. Really warming in red compared to ambient in blue. You see no effective warming on plant size in any of these populations, which is really interesting since you see a pretty drastic effect on phenology. And we often associate timing and size a lot in plants. And then I'll just show you some fecundity data. This is the number of seeds that they made. It's pretty striking. In the ambient treatments in blue, you see this decline, but really with increasing origin, right, of these populations from the site that they were planted. In the heated treatment, we see a recovery of fitness for most of these populations. And, you know, even the North Carolina population was able to eke out some seeds by the end of the growing season. I like this data, this is kind of a side point, because I think in the literature, I often see people measure plant size as an estimate of fecundity. And you can see those don't match well in this study, or really any study where time is a really important factor, and you potentially don't have close alignment between plant size and phenological time. So anyway, this seems like an obvious finding, right? You bring plants from way south up north, and they're going to flower or produce seeds so late in the season that you're not going to get anywhere with them. But again, it was just like proof of practice. What is practical to do with these assisted migration programs and what is not? I mean, bringing the most dry and warm adapted population up north is really only going to go so far, depending on when you're introducing those genotypes into that northern site. I wanted to look at assisted gene flow at the same time. We've heard a talk now just before about how important gene flow can be to populations that are struggling to adapt. So this is hypothetical data, and I wish I had a laser pointer here. Actually, can you see my mouse? No. All right, so what I'm going to show you here is hypothetical data on flowering schedules for these plants. So on the x-axis, you see day of the flowering season. Okay, day of the flowering season on the x-axis here, so short flowering season for this hypothetical plant. We have 2 populations that are flowering here and 3 individuals from each one. So these curves are just showing you the proportion of that individual's flowers that are open on any given day. And I have what I'm calling the resident population, maybe local to a site. And then individuals from a population that you maybe brought in an assisted migration program. So I'm calling them migrants in this case. And you can notice comparing these 6 flowering schedules, there's variation in flowering time and when they are flowering. Some of them overlap and some of them don't overlap at all. Another way to look at this is to think about the proportion of pollen available on any given day that come from these migrant individuals. And in the beginning of the growing season, that proportion is 0. There's a small window where there's increasing proportion of pollen from those migrants. And then at the end of the season, it's all from the migrants, right? So if you're thinking about what are the chances that these populations are going to hybridize and perhaps bringing in migrants might inject some really needed, valuable genetic variation into that resident population. Phenology can limit that. Again, this is hypothetical data, and I'll show you real data in a second. But if you look at the hybridization of these residents, so think about all the ovules in that resident population, and what proportion of those ovules are potentially pollinated by migrant pollen, it's 0 in the beginning of the growing season, and it increases a little bit for those later flowering individuals, but it doesn't reach, 50-50, which you might expect if the flowering schedules were perfectly overlapped. In the reverse scenario, these are the migrant ovules, and what are the chances that those will be fertilized by pollen from the resident individuals? Only those very early flowering migrant individuals will receive pollen from those resident individuals. So despite however many individuals you bring in, the individuals who are able to participate in hybridization are a subset of all the individuals there, and they're a biased subset. They're the ones that are most phenologically similar to each other. So we see biased gene flow, and it's actually asymmetric for a reason I can chat about if anyone is curious. So remember the shape of these curves, right? And what for the resident population here and the migrant population here. I counted flowers on my plants every day. I can chat about why I did that. At the time, I'm not sure that I was sure why, but I did do it. And here's what our data looks like. If you consider the Minnesota population, that northernmost population that's from the same latitude as my field site, as the resident population, I technically have 3 sort of assisted gene flow scenarios I could use to look at the chances of hybridization. And the shape of these curves is similar to what we saw in the previous slide, right? This is under ambient conditions. For the resident population, you are only going to see hybridization maybe for the latest flowering individuals and only from that next northernmost population, that Pennsylvania population. In the reverse, here's the migrant individuals. The migrant individuals, and it's only the earliest flowering individuals really from the 2 northernmost populations that you're bringing in that have a chance to hybridize. North Carolina, you're out of luck. Do the heated treatments or does future conditions actually change this? Not really. These graphs are almost identical except for maybe 2 North Carolina individuals that kind of got lucky down here. So my point here with this data, of course, we know that if there isn't good phenological synchrony between individuals, you're going to get a limited opportunity for gene flow between them, right? But again, with people proposing assisted gene flow and assisted migration as these, maybe surefire ways of conserving species, we found some pretty basic, like foundational biological things that we've known about for a while and demonstrated this could limit the success of your programs, right? I'm going to summarize what we found here. Latitudinal clines and phenology can determine the reproductive success of individuals in assisted migration programs, especially across latitudes where photoperiods might be important. Latitudinal clines can also limit the extent of hybridization between populations. And this is important because it could stall the effectiveness of assisted gene flow programs if this gene flow is biased in terms of who's participating and then asymmetric across those populations. So if you're expecting evolutionary rescue, it might take many, many times longer than you project than under like a random mating scenario. So when conservation practitioners are selecting candidate populations for either program, they have a lot of things they need to consider, including where the population is coming from, where are they going, how much do those climates match, including photoperiod, not just the variables that are changing with climate change, right? And also, when are you planning these conservation measures? There are a lot of people saving seeds right now that are planning maybe conservation measures 50 years from now. And so does my heated treatment perfectly mirror the conditions we might expect in 50 years? Maybe a little bit, but my results differed between my treatments, right? So if you're collecting genotypes now for conservation later, you really need to think about that difference, that temporal difference as well, and what you're bringing into these conservation programs in the future. All right, so that was a bit of what I did in grad school. And then I went on to do a postdoc at the University of Georgia, and I really wanted to study within the range dynamics and look at local adaptation. So I did that using Bucra stricta as a model. Looks like Arabidopsis. It is a mustard species. It's related to Arabidopsis. It's found in the Rocky Mountains. So I made this figure here just looking at herbarium specimens. It's predominantly selfing, and it will reproduce over multiple years once it gets established. It's small, which means that it's easy to grow and easy to transport and move around in space. So prior to my joining the lab, Jill Anderson had collected seeds from over 50 populations of Bucra stricta in the Rocky Mountains of Colorado. So here's a map of central Colorado. Here is an aerial view of the area. The Rocky Mountain Biological Laboratory is right around here. And all of these white dots are showing you where she collected seeds from these populations. And they look, she collected them across an elevational gradient, and it's kind of hard to see elevation here. So I'll show you here in a histogram. The royal we, really captured the elevational range of this species in this area, from where its lower elevation range boundary to higher elevation range boundary occurred, and then a bunch of populations in the center of the range. She had also established 5 common gardens across this elevational gradient here, from the lowest to the highest elevation. So this really set the stage to study local adaptation, to study the effects of climate, and try and do so in a really thorough way with 50 populations. And quickly, I'll say that elevational gradients are excellent proxies for climate. If you look here at these 3 panels, elevation is on the x-axis of all of them. Each of these black dots is a population that we collected from. The red dots are the 5 common gardens, and you can see how well elevation scales with 3 important measures of climate in this area. We've got mean annual temperature on the left, and mean annual precipitation in the middle, and then an aridity index on the right. So elevation very closely approximates climatic gradients in this area. And in terms of climatically important variables for climate change, we all know temperatures are rising everywhere, and that is true of this region as well. This red vertical bar here is showing you at the time of our study how much warming had occurred, and then you can see how much more is expected. I should say this is winter warming. On the right is a variable we might think about a little less, but is hugely important in this region, and that's snow depth or snowpack. Over the past decade or 2 in this region, we've seen drastic decreases in the amount of snow that's fallen. And that's expected to continue to decrease to the point where, like, there's entire industries out there, skiing, snowboarding, or whatnot, that are in danger of failing entirely. There are years where we have like no wildflowers at all out there because there's no snow to insulate the plants, and those flower buds all get frost damage from the cold temperatures of the air, and then nothing blooms. So that's another big tourist attraction to the area. So here's the experimental design in cartoon form. Here's our elevational gradient, the big rectangle. And I'm showing you 5 populations for now, but we had 50. So 5 populations, and consider those our common gardens. And what we did was reciprocally transplant all 50 populations across all 5 of those gardens. And then we also imposed an artificial snow removal treatment in those gardens to mimic the conditions that we would experience under climate change. And by that, I mean literally shoveling snow out of plots to mimic the reduced snowpack that we would experience in the future. I'll show you a video of that in a second. For this paper, we had a little over 100,000 seeds and juvenile rosettes. We plant both life stages out into the field for this study. And I think by now, because of there's been several years that have passed, there's probably over 200,000 plants that have gone into this study. I want to show you what these gardens look like. This is the lowest elevation common garden on the left in August, and this is the highest elevation one in the right in August. That's Jill Anderson right there taking some data. And wow, do they look different? You wouldn't think that you could drive from one site to the other site in a pretty short amount of time. So elevational gradients are severe. Really hot and dry down at lower elevation, and moist and lush at higher elevation, although the growing seasons are shorter. I want to show you, I hope this works. Well, this is our mid-elevation garden. This is right by the field station. And I'll show you what you're looking at before I hit play. This is what it looks like after we go and shovel snow out of these plots. And for the high elevation gardens, we hire experts with avalanche training to go do this for us. But we have poles that are sticking out of the ground. It's kind of hard to see how far down this garden goes. It's quite large. But poles that are sticking out of the ground in opposite corners. So you always know where your plot is underneath the snow. And we shovel snow down to about 10 centimeters above the ground. And we remove it from the garden. And we completely circumvent any area that has control plots. So those remain completely unmanipulated. And so this video is going to start right after this treatment was applied. Is it going? No. I feel like we tested this. Hold on. How do I? Did this work for you, David? Are you here? Okay. Well, I can explain it won't be as exciting, but I will try my absolute best. Okay. Here's what you would see if the video was playing. What happens within these plots is that the snow starts to melt really rapidly. In fact, within a day or two, you start to see all of our plant pads poking out of here. The snow melts out really rapidly here, like many, many weeks faster than you see it melting out over these controlled plots. And counterintuitively, what this does when there's no snow is it exposes those plants to frost damage events. They're very susceptible to frost because those daytime, nighttime temperature fluctuations are still happening, and it can really damage their tissues, especially their floral tissues. Snow is an excellent thermal insulator. So anything hanging out under the snow is not frozen. It's actually hanging out at about 4 degrees Celsius and quite protected. So these individuals that are in our snow removal plots are exposed to sub-zero temperatures. They're susceptible to frost damage. They actually receive less of that snow melt than the plants in the control plot. So it's kind of a multi-variable manipulation. We take a lot of data on these plants. Here are the traits that are important for this particular set of results. So every year we go in and assess whether individuals are alive from the previous year. We also measure their size so we can monitor growth over time. So this is an iterative thing for as long as a plant is alive. Some years these adults will go reproductive. So we measure whether they made flowers, whether they made seeds, and a measure of how many seeds they made. We plant a bunch of seeds out there to get germination data, and also monitor the seedlings to get data on establishment and whether they recruit into the sort of adult portion of the life cycle. And we've also done some dormancy assays. So a fraction of seeds, it's the big black box of the life history cycle, right? What happens to those seeds once they're dispersed. So we've done some seed dormancy assays just to look at the rate at which seeds actually germinate out of the seed bank, and then how many years that can persist for. So these are the traits that are relevant for this particular study for now. And it's because we used all of these, I'll call them vital rates, in an integral projection model. So I'm happy to answer questions about this after the talk, but we used an integral projection model to forecast population growth rates for each of our populations in both treatments, the ambient and the early snow removal. So these models had treatment, source elevation, also the elevation of the garden. And then this was a size-based integral projection model. So plant size is really what's dictating the progression through these stages, and also the, for instance, the degree of fecundity that a plant might have every single year. Here's a layout of this graph before I show you the results. We have population growth rate on the y-axis. So 1 is indicative that the population is able to replace itself. Anything below 1 is dire news. On the x-axis is the elevation of the garden. That's not exactly linear, but it's close enough. So low to high elevation. What I want to show you first are these growth rate parameters for the local populations to the gardens. So like, let's look at local adaptation, right? This is what they look like. Yikes, right? These three, we actually see that in all cases, except I guess I should say in blue, that's our ambient treatment. And in pink, that's the snow removal treatment. And in all cases, except the snow removal treatment in one garden, all of those population growth rates and their confidence intervals are below replacement. Like range wide, at least elevational range wide maladaptation in this system. This was shocking to us. Also shocking that our snow removal treatment had an effect on individual vital rates, but in terms of the population growth rates, not really. The biggest effect had to do with where they were along this elevational gradient. Okay, so that didn't look so great for this species, but what happens when you transplant all these populations into these gardens? Do we see anyone doing any better anywhere? And again, I'm just going to focus on the local populations, although we had 50 in each site. Here's the low elevation garden. Nobody likes it there. It's really sad. In fact, we've seen, since Jill collected those seeds, we've just seen all the local populations at low elevations go extinct. Like before our eyes, that range is retracting upslope. Okay, next garden, still bad, especially the northern population. High elevation population does not like going down slope. Mid elevation garden, second highest elevation garden, still nobody up there. And then the highest elevation garden, where finally, not the local population, but one population does okay there under ambient treatment. So this was really shocking to us. Widespread, kind of ubiquitous maladaptation. I want to rearrange this graph to emphasize one more point. Here, what I'm calculating on the x-axis is the difference between the elevation a garden came from, so the source elevation, and where we planted it. And the y-axis is growth rate. So at 0, if populations were locally adapted, at 0, you'd expect the highest population growth rate. You put a population in its home elevation, and you have the highest growth rate. And you should see that growth rate decline with increasing distance in either direction from that home elevation, right? So when we impose our data on this rearrangement of the x-axis, this is what you see. That decline is definitely happening here when you move high elevation populations to low elevation sites. But when you move low elevation populations to higher elevation sites, again, no one's really above 1 here, but they're doing better. And this suggested to us that for those low elevation genotypes to persist, they need to move upslope. Hopefully give them a little bit of time to adapt, maybe not introgress. I have learned I should use that word to talk about between species gene flow, to hybridize with the populations that are higher upslope, and hopefully we'll see some adaptive evolution occur. Okay, one last component to this study that I think is really cool and I want to share with you. We saw maladaptation range wide and we weren't expecting it. We wanted to see whether or not we could figure out what's causing that maladaptation. So we decided to impose a snow addition treatment. It's like the opposite of the snow removal treatment. We had these barrels, we cut the bottoms off, and I now see I'm late on time. So we flipped them upside down and we focused on the seedling stage and the germination stage because you can fit a whole lot within these barrels. We added snow to them. They would accumulate snow. We would just keep adding snow. So we had delayed snow melt like you might expect maybe 30 years prior. And we wanted to see if we could recover evidence of local adaptation, at least at that life history stage, which would then let us know it's climate change and specifically the snowpack dynamics that are contributing to that pattern of maladaptation that we found. Wow, that's like missing. Anyway, the short story is under the control and snow removal treatments, pink and blue, again, we see this pattern where the local populations don't have the highest germination success. But here in black, this is our snow addition treatment. We were able to recover, at least for germination anyway, that signature of local adaptation, which lets us identify a culprit to the maladaptation we were seeing. So local maladaptation is widespread in our study system. Climate change is the likely cause, at least for one life history stage. That's a very vulnerable one. And upslope migration is favored if those low elevation populations have any hope of sticking around. I can make this quick. Snow loss is happening everywhere. Here we go. Okay. Natural upslope migration is very unlikely in this species because it's got really short distance seed dispersal. Gene flow between populations is really unlikely. It's highly selfing. We have documented clines, elevational clines in flowering. So we've already talked about phenological asynchrony and what that means for hybridization. And they almost no individuals reproduce each year. So the point of this graph, if you just look at the y-axis here, proportion of individuals that flowered, this was a good year, okay, 1%, 3%, maybe as high as 15% for one population in 1 year, but it's low, like these individuals are struggling. All right, really quick. Here is the grant that my collaborators and I just got funded. I think you'll see how nicely my previous work fits into this work that we're starting now. Here's my collaborators. Just collaborate with awesome people who are friendly and amazing. Emily Josephs at MSU, Jill Anderson and Megan DeMarche at University of Georgia, Seema Sheth, who is collaborating with like half of the speakers today, I think. And Jenny Cruz Sanders from the Georgia Botanic Garden and me. And this is the question we want to ask. To what extent can we improve predictions of range-wide dynamics under climate change by identifying the mechanisms that impact populations throughout the range? Here are some of those environmental and eco-evolutionary processes that we know are impacting populations in unique ways. And what we want to do is measure them, quantify them, and see if they're explaining population variation in responses to climate change, and then project range-wide dynamics under climate change. So aim one, we will collect tissue from populations throughout the entire range that's happening this summer in order to estimate rates of historical gene flow and look for genomic evidence of adaptation. In aim two, this will be a manipulative experiment in 4 common gardens. That's where these triangles come in, 3 within the range, and then 1 back out in Ontario beyond the range. We will assess for 12 populations in those sites the degree of local adaptation. We will conduct paternal half-sib crosses in order to be able to estimate heritability and adaptive genetic variation for fitness and related traits. So look at adaptive potential and also assess phenotypic plasticity. Within each site, we're going to experimentally warm plants using these tall open top chambers that can warm temperatures within for tall plants in big communities. So we've tested that with a different set of materials. So here's a summary of what I just said. We'll measure phenology, growth, stress, survival, reproduction. We'll also assess dormancy and estimate dispersal kernels for these populations. And ultimately, what we want to do is use this data in a bunch of different kinds of models, many of which you've heard about today. So like if you have questions about genetic offset models, you can ask Daniel because he's here. So all of these different models and then a few more, we want to know what data is actually needed to make informed predictions about responses to climate change. It would be great if conservation practitioners really didn't need to know the heritability of some traits in order to predict how populations will respond to climate change. So these are some of the models that we'll focus on. And then lastly, we are collaborating with a few different conservation groups in order to build a tool for them based on their needs and input that will let them forecast population and range-wide dynamics under climate change with a variety of data they may or may not have, or they may or may not be able to get from the literature to help them with their conservation management plans. Okay, I'm out of time, but I can talk about these other projects as well. And thank you very much. I'm happy to take questions.
Do we have questions? Who's in? First of all, thank you for your talk. It's really, really interesting and very thorough. Second, I study monkeys, so I'm sorry if I say something that just doesn't make any sense. But what I was thinking is, I had 3 questions that are somehow related to the first part of the talk and probably to this last part of the talk, and is, so what is the divergence of these populations genetically, how distinct they are? And I'm very skeptic of this moving things around by humans. So if you're thinking about introgression, and depending on how distinct these groups are genetically, it might be that you are creating monoweb hybrid things, for example. So at what extent can you do first, like in vitro reproduction to test whether this kind of combinations, genetic combinations are possible, and then maybe instead of moving plants, moving those that are making experiments with those that you know the genetic combinations. I hope it makes sense.
Yeah, like using the hybrids instead, maybe, or some, yeah, so a couple of things in there that were great and super relevant. How genetically diverged are these populations? I mean, I tend to focus on phenotypes, which is why I'm collaborating with people that focus on genotypes more than I do. I see a lot of phenotypic diversity maintained in the across sites where I plant them and in the greenhouse. So at least I know phenotypically they're quite diverged. Part of this grant, okay, well, wow, okay. This is, anyway, part of the grant, the first aim of the grant is to do exactly what you're suggesting and to assess how differentiated these populations are, how connected they are via gene flow, the extent to which they're differentiated due to responding to local selection pressures versus drift or some other thing. So that's something that we're doing in the current grant on that same species, which will feed back to my prior work, which is really exciting. You asked about hybrids. This is not me, this is Jill Anderson for the mustard species. She's hybridizing populations from across that elevational gradient. It is super unlikely to happen naturally, like I was telling you all. She's making it happen, and we'll put hybrids out in the field. How much can conservation practitioners do that? The answer is 0. They cannot. They either the species or the population is rare and they just don't, and they can't afford to provide seeds for experimentation, or they don't have the time or resources and to do something like that. And so they rely very heavily on us to come in and do experiments like this. Do I think that we'll make hybrids on the Chamaecrista? Maybe. It would be a side project. It's not the sample size is ballooning before our eyes, and we just can't have hybrids with pedigree population like we're doing in this experiment. But it is absolutely a natural step, especially when you have phenological asynchrony like we keep seeing, right? The solution there would be to hybridize them first and perhaps put hybrids out in the field, or where you're seeing low elevation or low latitude populations go extinct. Try and save some of that genetic material, separately, but also in hybrids, and maybe help that gene flow that wouldn't happen naturally. So I don't know if I answered all your questions, but
yeah, more than anything, not only that they are this gene flow, but it's not a hybrid sync effect.
Yeah. So you could do common garden or reciprocal transplants and actually see, and try and do it in a responsible way to where you weren't introgressing into the natural population, and try and assess whether or not those hybrids are actually fit or not. Crucial step.
Super awesome talk. I had a question about how generalizable you think those mustard results are to the rest of the community. Obviously, all plants have really different traits. But do you think we're seeing widespread maladaptation in these plant communities, and what are the potential effects of that?
Yeah, so I skipped this slide, and it maybe should not have, because that was the point I was going to make here. These are snow monitoring stations for the Intermountain West, and any red dot is a net loss in snowpack, right? Snow these snow dynamics are so important for all of these alpine, subalpine communities that we would be shocked if people, I mean, not everyone is studying everything, but we suspect that widespread maladaptation is so much more common than just our one species. So I think it's pretty widespread, especially in mountainous regions. It's just not, it's not very optimistic, is it? Yeah. Mhm.
Okay, I just have a few, thanks Susanna one more time. And I'll just yell, but I just have a few quick thank yous to say before we head to the poster session. Okay, so first of all, I want to thank our excellent admin staff who are enjoying the nice weather outside. Justin and Andy, thank you so much to the committee for everything that you've done. So David, Mia, Rosie, Abby, Gina, and Heidi. Thank you. Thank you to the past CLS and EEB for funding, especially Gina, who helped raise money to make this happen. Thank you to everyone who came here for this. We have at least 5, and I think more institutions represented among the attendees, and some of those people we'll get to hear about in the posters. Like for instance, some folks from undergraduate only institutions who came here and getting to give posters. We're really excited you're here and we get to talk science with you. So thank you for coming. And then finally, I'd like to thank our speakers one last time. It's such a pleasure to hear all of your excellent research, and it's such a cool event that we get to hear from up and coming scientists and have these keynotes. So thank you for flying in and traveling in to talk to us. Now there's going to be some logistics.
Okay. The posters are all set up. When we break from here in just a moment, the posters are in the conservatory. They are in no order. That is, it's whatever was the delivery order. So you, the presenters, will have to find it. Just walk down the main aisle. Same with everybody here. There will be food and refreshments. We are a public interface of the university, so the alcohol has to stay in this room because the public is still out there. The university does not allow its liquor license to be used in a public venue. So because we're open until 4:30, keep the beer in here. After 4:30, I'm not policing. Those were all my logistics.
There's one more logistic, which is the last shuttle. The last shuttle leaves at 5:30. So if you need to get on the shuttle, that's important. All right, enjoy the poster session and the reception. Thanks, everyone.